So in 1799, European scientists were handed the body of a platypus, and they were so profoundly confused by what they were looking at that they assumed it was a literal
Right,
hoax.
like an actual joke.
Exactly. They genuinely believed a prankster or like a taxidermist had just sewn a duck's bill onto a small mammal's body. Today, obviously, we know the platypus is very real. But that initial reaction, I mean, it proves a really fascinating point. Nature just absolutely refuses to fit into the neat, organized little boxes that humans try to build for it.
Oh, totally. And that tension is really the entire driving force behind taxonomy. We have this deep, almost hardwired human desire to categorize our environment,
Mhm.
to draw clean lines and establish order so we know exactly what we're looking at.
Right.
But biology is just this continuous, messy, unbroken chain of evolutionary changes. So drawing a strict line between one group of living things and another is, well, it's often like trying to draw a line in the ocean.
Which is the core mission of this deep dive. Today, we are exploring the classification of living things. And for those of you listening who are prepping for nursing entrance exams like the TEAS or the HESI, this is highly relevant, high-yield material. You really need to know these concepts.
Absolutely. But we're not just going to read flashcards at you today.
No, definitely not. We are looking at the tree of life and humanity's roughly 2,000-year quest to map the whole thing out. We're going to explore how an obscure microbe found in a boiling hot spring just overnight rewrote biology textbooks, and why, technically speaking, every pigeon you see on the street is a highly evolved reptile.
It is a wild ride.
Yeah.
And to understand how we map that tree today, you really have to look at the tools that early thinkers had at their disposal. For nearly 2,000 years, Western thought was anchored by Aristotle, around 384 BCE.
Wow, so going way back.
Yeah, way back. And he built a systematic biological framework using the only instrument he actually had, which was his naked eye.
Right. He looked at the natural world and grouped animals by obvious physical traits, like blooded versus bloodless, which, interestingly enough, actually maps reasonably well onto what we now call vertebrates and invertebrates.
It's not a bad starting point, really.
Yeah.
He also proposed the great chain of being, which was this strictly linear ranking of organisms. It went from the simplest things up to the most complex, with humans sitting comfortably at the very top, of course.
Naturally. And that linear visual framework held strong for centuries, right? I mean, it works fine when you are only trying to categorize, say, the local flora and fauna of the Mediterranean.
Exactly. But fast forward to the 1700s, and global exploration is in full swing. Naturalists are suddenly shipping thousands of entirely new, totally alien-looking plants and animals back to Europe.
And the old system simply collapsed under the weight of all this new information. I mean, the naming conventions devolved into total chaos.
Oh, it was a nightmare. A single plant's official scientific name was essentially a polynomial. It was a Latin phrase that could run 10 to 12 words long, describing every single leaf and petal.
So if you were a physician trying to reference a specific medicinal herb, you basically had to memorize a paragraph.
Yeah, and to make matters worse, a botanist in France might use a completely different descriptive paragraph than a botanist in England for the exact same plant. There was zero universality.
Okay, let's unpack this. Imagine the early days of the internet, right? Right. But there are no search engines, no standardized URLs, and everyone just saves their files to one massive, shared, unorganized desktop.
That sounds terrifying.
Right. People are using incredibly long, highly specific file names that no one else agrees on. It would be a totally unusable disaster, and that was biology in the early 18th century. But then into this disaster steps Carl Linnaeus.
Yes, Linnaeus. He was a Swedish botanist who decided to build the ultimate folder system for life on Earth. In 1735, he published a relatively short pamphlet called Systema Naturae.
And that changed everything.
It really did. By the end of his life, he had expanded it into massive multi-volume editions that classified roughly 10,000 species. And he achieved this using two structural inventions that are still the absolute bedrock of taxonomy today.
So let's talk about the first one-nested hierarchical ranks. You don't just have one massive scrolling list of animals. You create categories and then subcategories within those categories.
Exactly. Linnaeus started with kingdom, class, order, genus, and species. Later, scientists realized we needed a bit more granularity, so they added phylum and family into the mix.
Right, and his second invention completely revolutionized how scientists communicated. It's called binomial nomenclature.
Yes, formalized for plants in 1753 and animals in 1758. This system dictates that every single recognized species on the planet gets a unique two-part Latin name.
the genus, which is capitalized, and the specific epithet, or the species name, which is always lowercase. And both are italicized, right?
Spot on. Suddenly, those 12-word descriptive paragraphs were gone, replaced by these really elegant, scalable two-word tags.
But Linnaeus built this massive, world-changing architecture using that exact same ancient toolset Aristotle used. He was just looking at things.
Right, he used a phonetic approach. He grouped organisms based purely on visible similarities. He counted the reproductive organs of flowers, or he looked at the shape of an animal's toes.
And when your entire methodology relies on trusting your eyes, you're definitely going to make some pretty significant miscalculations.
You're going to be fooled by superficial resemblances. Like, in the very first edition of his book, Linnaeus classified whales as fish.
I mean, I guess if you're just looking at it.
Yeah, he observed an aquatic animal with a streamlined body and fins and just filed it in the fish folder. He did eventually correct this in later editions, moving them to mammals once he focused on the fact that whales have a four-chambered heart, are warm-blooded, and nurse their young.
He also caused a massive cultural uproar with a group he named Anthropomorpha. He put humans, apes, and sloths all in the exact same folder based on their physical anatomy.
Which, in the 1700s, suggesting there was no distinct biological separation between humans and animals, was seen as incredibly controversial. Honestly, heretical in some circles.
Yeah, but Linnaeus was simply following his visual data where it led him, regardless of the cultural lens of his time. And despite those early visual errors, the structural framework he built was just so robust that it survived.
It adapted.
Right.
That structure really is the backbone of modern biology. And if you were taking an exam, you definitely need to know the exact sequence of this nested hierarchy from broadest to narrowest.
Okay, let's run through it. We have domain, kingdom, phylum, class, order, family, genus, and species. The classic mnemonic to remember this is, Dear King Philip came over for good soup.
A lifesaver for exams, truly. And the defining mechanic of this hierarchy is that it is strictly nested. There's no overlap whatsoever.
I always like to think of Russian nesting dolls. The order doll fits perfectly inside the class doll, and it completely contains the family doll.
That's a perfect way to picture it. If two organisms are in the same family, they must, by definition, be in the same order: class, phylum, kingdom, and domain.
Let's trace that path for ourselves, just to see it in action. Humans are Homo sapiens. We sit in domain boner. Eukarya, meaning our cells have a nucleus.
Yep.
We are in kingdom Animalia, phylum Chordata, class Mammalia, order Primates, family Hominidae, genus Homo, and species Sapiens.
Now, if you run that exact same trace for a chimpanzee, Pan troglodytes, you will find they share every single nesting doll with us.
Wait, really? Every single one?
Up until the very end, yeah. Eukarya, Animalia, Chordata, Mammalia, Primates, Hominidae. The evolutionary road only forks at the genus level.
Which is such a beautiful reflection of our actual genetic reality. That split at the genus level aligns with modern genomic sequencing, showing we share roughly 98 to 99% of our DNA with chimpanzees.
Exactly. It maps perfectly to a shared common ancestor from about 6 to 7 million years ago. The folder system isn't just arbitrary. It actually tells the story of our evolutionary history.
And that storytelling extends to the binomial nomenclature, right? Like, a common name like Daisy is totally useless in a scientific context.
Completely useless. It could refer to completely different plant families depending on whether you were in North America, Europe, or Australia. But the Latin binomial Bellus perennis identifies one specific evolutionary lineage, and it means the exact same thing to every botanist on Earth.
And that universality is why the medical field relies entirely on this system. Nurses and clinicians don't use colloquial names for pathogens on a medical chart.
No, definitely not. You're right. Staphylococcus aureus or Plasmodium falciparum, it leaves absolutely no room for ambiguity.
A perfect example of this is a bacterium everyone is familiar with, Escherichia coli.
Oh, E. coli, yeah. The binomial name Escherichia coli is incredibly descriptive if you know how to read it. It was named after Theodor Escherich, a German pediatrician who first isolated it in 1885 while studying the intestinal flora of healthy infants.
So the genus Escherichia immortalizes the scientist.
Exactly. And the species, coli, directs you straight to its natural habitat, the colon.
So a nurse in Seoul, a lab technician in São Paulo, and a physician in Toronto can all look at that name and know exactly what organism they're dealing with, its cellular structure and its habitat, all without any translation software.
It's a brilliant global language. But we have to remember, this entire system relied for centuries on anatomy, on form, and relying on an anatomy chart can be deeply misleading.
Right, because look at a shark and a dolphin. They are both sleek, gray, streamlined apex predators with dorsal fins and flippers. If you only look at their form, you put them in the same folder.
And that highlights the critical difference between the phonetic approach of early taxonomy and the phylogenetic approach we use today.
Phylogenetics groups organisms strictly by their evolutionary relationships, who shares a common ancestor.
And sharks and dolphins do not.
Not recently, no. Sharks are cartilaginous fish. Dolphins are mammals with lungs and a placenta. What you are observing there is convergent evolution. They independently converged on the same body plan because, well, physics demands it. If you want to move fast through water to catch prey, a sleek torpedo shape with fins is just the best engineering solution.
Exactly. The environment shaped them identically, but their underlying genetics are worlds apart. So to stop being fooled by convergent evolution, taxonomy had to move past the microscope and start looking at the genetic code itself.
Which brings us to the mid-20th century. We started finally overhauling the Linnaean system. In 1969, Robert Whittaker proposed a five-kingdom system.
Right, because up to that point, fungi and bacteria were awkwardly shoehorned in with plants, which makes no sense. Whittaker separated them out into Monera, Protista, fungi, plantae, and animalia.
Mostly based on their cellular organization and how they get their nutrients right.
Yes. But here's where it gets really interesting. Whittaker's system was a massive improvement, but it was still just a prelude to the real earthquake in 1977.
Oh, Carl Woese.
Yes. Carl Woese, a microbiologist who decided to map the tree of life using a completely different method. He didn't care what a cell looked like or what it ate. He looked at ribosomal RNA, or rRNA.
And this is a really crucial mechanism to understand for the listener. Why rRNA?
Well, ribosomes are the protein factories of the cell. Every single living thing on Earth has them. Because they're so essential to survival, the genetic code that builds them mutates incredibly slowly.
Because if a mutation breaks the ribosome, the organism just dies instantly.
Precisely. Therefore, rRNA acts as this highly conserved, universal molecular clock. By comparing the RNA sequences of different organisms, Woese could mathematically measure how long ago they shared a common ancestor.
So Woese started analyzing the RNA of various organisms, including these bacteria that produce methane, and the data he got back completely shattered the foundations of biology.
It really did. Prior to 1977, the scientific consensus was just that all bacteria were essentially the same thing, simple single-celled organisms without a nucleus.
But the molecular clock revealed that these methane-producing microbes were not bacteria at all.
Not even close. They represented an entirely distinct lineage of life. They were as genetically distinct from regular bacteria as a human being is from a bacterium.
That's just wild to think about. They just shared a similar superficial shape under a microscope.
It was a staggering discovery.
Yeah.
Woese actually faced immense pushback from the scientific community. A lot of scientists simply couldn't accept that their visual categorizations had been so fundamentally wrong for so long.
But the data won out. It ultimately forced the creation of a brand new taxonomic rank, sitting even above the kingdom, the domain. We now operate on the three-domain system: bacteria, archaea, and eukarya.
And to understand just how profoundly this hidden genetic world impacts our daily lives, we have to look at Thomas Brock.
Yes, in 1969, Brock was researching in Yellowstone National Park and discovered a microbe living in the boiling hot springs. We're talking water around 158 degrees Fahrenheit.
Right, and he named it Thermus aquaticus. Initially, it was just cataloged as a fascinating, heat-resistant bacterium.
But then our understanding evolved.
Yeah, the subsequent understanding of extreme microbes, many of which belong to this newly discovered archaea domain, or our deeply ancient bacterial lineages, reveal that their internal biochemistry is fundamentally different from normal life.
Their proteins and enzymes have actually evolved to remain stable at temperatures that would literally cook and disintegrate human cells.
And that heat stability led to one of the greatest technological leaps in medical history.
The PCR test. When scientists wanted to copy DNA in a lab, a process called polymerase chain reaction, or PCR, they had a major problem. To separate the DNA strands, you have to heat them up almost to boiling.
But that heat naturally destroys the enzyme needed to copy the DNA, meaning scientists had to manually add new enzymes every single cycle. It was excruciatingly slow.
Until they isolated Taq polymerase, the DNA copying enzyme from Thermus aquaticus. Because this microbe lives in boiling water, its polymerase easily survives the heating cycle of PCR.
It allowed scientists to fully automate the DNA amplification process.
So an obscure microbe pulled from a hot spring became the engine for modern genetic sequencing, forensics, and literally every single COVID-19 PCR test run globally. It is the ultimate proof that accurate biological classification isn't just academic bookkeeping. It unlocks the operating manual of the planet.
Beautifully put.
But I have to push back here for a second.
Oh, go for it.
If we have this incredible tool now, if we can just sequence RNA and DNA, why isn't this field finished? I mean, I can open any biology textbook right now and find fierce ongoing debates about the definition of a species. Shouldn't the molecular data just hand us the final answer?
What's fascinating here is that a species is largely a human construct, while evolution is a fluid process. Nature doesn't care about our neat little boxes.
So the definition itself is the problem.
Exactly. The most widely taught definition, especially for exams like the TEAS or HESI A2, is the biological species concept, formalized by Ernst Mayr in 1942. It defines a species as a group of populations whose members have the potential to interbreed in nature and produce viable, fertile offspring.
Meaning they're reproductively isolated from other groups. If they can naturally have babies together, they are a species. It is an elegant definition when you are looking at, say, lions or elephants.
But it completely collapses when you zoom in. Consider the millions of microbial lineages that reproduce asexually. Bacteria just replicate their DNA and divide in half. There is zero interbreeding.
So if you strictly apply Mayr's concept, every single individual bacterial cell is technically its own distinct species.
Which is obviously unworkable. It also struggles with the messy realities of the animal kingdom. We have ligers born from lions and tigers and mules born from horses and donkeys. The boundaries are highly porous.
My absolute favorite example of this breakdown is the phenomenon of ring species, like the Ensatina salamanders in California.
Oh, ring species are perfect for this. They showcase evolution happening in real time across geography. Picture the Central Valley of California. Millions of years ago, a population of salamanders at the northern end of the valley began migrating south.
And the population split, right, with one group moving down the eastern foothills and the other moving down the western coastal mountains.
Exactly. As they move south, each population adapts to its specific local environment. But here is the catch. The salamanders on the eastern side can still breed with their immediate neighbors to the north and south.
And the western group can also interbreed with their immediate neighbors.
Right. But by the time these two lineages finally meet at the southern end of the valley in Southern California, they have accumulated so many genetic differences that they can no longer interbreed. The eastern and western salamanders act like two completely distinct species.
They are reproductively isolated at the bottom of the ring, but intimately connected by an unbroken chain of interbreeding cousins all the way back to the north. So are they one species or two?
There's no clean answer. The biological species concept just shatters. Taxonomists actually had to juggle multiple competing concepts: morphological, ecological, phylogenetic, depending on the specific organisms they are studying.
And if the definition of a species is messy, the structure of the tree of life is even messier. We are used to seeing these beautiful branching diagrams where a single lineage splits into two and they never cross paths again.
But when you look at the microbial world, it is not a tree, it's a web.
Because of horizontal gene transfer?
Yes. Microbes don't just pass genetic information down vertically from parent to offspring.
Right.
They can pass DNA directly across to completely unrelated lineages in their environment.
It's like trading a trading card, but with superpowers. A bacterium can acquire a plasmid containing antibiotic resistance and simply hand it over to an entirely different species of bacteria nearby.
It would be like a human shaking hands with an oak tree and suddenly acquiring the genetic instructions for photosynthesis. It makes tracing their evolutionary history incredibly tangled.
And that tangled reality extends to eukaryotes as well, right? For decades, textbooks included kingdom Protista, but Protista was largely a wastebasket taxon.
It really was. If scientists found an organism with a nucleus, but it didn't fit the neat definitions of a plant, animal, or fungus, they just tossed it into the Protista bin. Amoebas, giant kelp, slime molds.
It was literally the junk drawer of biology.
Modern molecular phylogenetics prove that Protista is not a natural group. It does not share a single exclusive common ancestor. It is highly paraphyletic, a massive collection of vastly different evolutionary paths.
Consequently, you will find modern curricula moving away from treating it as a valid formal kingdom.
Which brings us to the most mind-bending consequence of mapping the true evolutionary tree. In the mid-20th century, an entomologist named Willy Hennig pioneered cladistics.
And cladistics insists that any valid taxonomic group must be strictly monophyletic, meaning it must include a common ancestor and absolutely all of its descendants, with zero exceptions.
When you enforce that strict mathematical rule, traditional categories face major problems. Take the class Reptilia. Traditional taxonomy grouped snakes, lizards, turtles, and crocodiles as reptiles, but placed birds in their own separate class, aves.
But the molecular and fossil data show definitively that birds are the direct descendants of theropod dinosaurs. They branched off from within the reptile family tree.
Which means if you define reptiles as a group that excludes birds, your group is incomplete. It's paraphyletic.
Wow.
To maintain a scientifically valid monophyletic group, cladistics demands that birds be classified as living dinosaurs, and therefore part of the broader reptile clade.
So what does this all mean? We have journeyed from Aristotle's bloodless animals to Linnaeus's chaotic folders, all the way to boiling microbes and feathered reptiles. But for the listener trying to digest this for their exams and their daily clinical practice, what is the practical takeaway?
Well, if we connect this to the bigger picture, the distinction between these categories is quite literally a matter of life and death. The split between domain bacteria and domain archaea isn't just exam trivia. They have fundamentally different biochemistry.
Right, bacterial cell walls contain peptidoglycan, and their ribosomes have specific structures. Archaea lack these entirely.
Which dictates medical treatment. Many of our most common antibiotics work by specifically targeting and destroying that bacterial cell wall, or gumming up the bacterial ribosome.
So if you misclassify an infection if you try to use those antibiotics against a virus, a fungus, or an archaeon, the drug will do absolutely nothing. You have to know the precise biological domain of your enemy.
It also forms the front line of global public health. When a novel pathogen emerges, such as a new coronavirus, rapidly classifying it into its proper viral family allows epidemiologists to predict its behavior.
They can anticipate how it transmits, how quickly it mutates, and what vulnerabilities it might have, entirely based on the traits of its closest evolutionary relatives.
And the stakes are just as high in ecology. If conservationists discover that a specific regional population of whales isn't just a variant, but an entirely distinct, genetically isolated species, that scientific classification can instantly trigger international legal protections.
Or, in agriculture, managing an invasive species requires knowing exactly where it sits on the tree of life, so you can study its native ecosystem to find targeted, natural predators.
Taxonomy provides the map. If our map is inaccurate, our clinical interventions fail, our environmental policies fail, and our fundamental understanding of our place in the biosphere is compromised.
Let's bring this all together. Today, we tracked humanity's attempt to draw that map. We started with the purely visual observations of Aristotle, moved through the necessary but flawed structural genius of Linnaeus's binomial nomenclature, and arrived at Carl Woese,
who proved that the molecular clock of RNA is far more reliable than the lens of a microscope.
Exactly. For your exams, remember the core mechanics. Keep your nested hierarchy straight, domain down to species. Understand that a binomial name provides global clarity.
Recognize that convergent evolution can trick the eye, making DNA the ultimate arbiter of ancestry. Know that Carl Woese's molecular data birthed the three-domain system, separating bacteria and archaea.
And finally, remember that while the biological species concept is a useful tool, it has distinct limitations, particularly with asexual organisms and ring species. Classification is really a human attempt to impose digital binary logic onto an analog continuous world.
The tree of life is not a static poster you just memorize. It is a dynamic, shifting puzzle. Every time we sequence a new genome or pull a bizarre new microbe from a deep-sea hydrothermal vent, the map gets redrawn.
It's an ongoing pursuit of truth that constantly forces us to abandon our old assumptions. We spend so much time figuring out how to trace these branches backward to find common ancestors. But I will leave you with this final thought.
Oh, I like where this is going.
Right now, synthetic biologists are utilizing artificial intelligence to design entirely novel genetic sequences from scratch. They are building synthetic organisms in laboratories that have absolutely no evolutionary history.
Which completely breaks the model.
Exactly. When an organism is born today, engineered from the ground up, with no ancestors and no evolutionary lineage to trace back through the tree, how do we classify it? What box do we put it in when it breaks the fundamental rules of life itself? Thanks for taking this deep dive with us. Keep questioning the boxes.